one of the things we're doing is if anyone wants to take a picture of that QR code, you can actually make suggestions about a future topic. So we have these every 2 months. And again, the purpose of these is to bring together academics, clinicians, care providers, and the local industry environment. And so what we aim to do, like today, is have someone present the scientific perspective, the clinical perspective, and then maybe two related biotechs sort of in the related areas that may be working in or around that general area. So we're always welcoming new topics. So the one we're doing the next one we'll be doing in April is around regenerative medicine. So we haven't chosen, figured out speakers of the companies yet, but there's a lot of people to choose from in that area in Vancouver. But yeah, welcome to any topics. Okay, so I think we'll start with the conference now, the workshop. So welcome everyone to our first Discovery to Commercialization event of 2021. We got a great list of speakers today, and I thank all the speakers for joining on today. And before I begin, I'd just like to acknowledge that we're conducting this business, at least from the VCHRI standpoint, from the shared traditional homelands of the Musqueam, Squamish, and Tsleil-Waututh peoples. Next slide. So these are, as I mentioned, these are the dates. To save the date, we don't have the exact date yet for April, but it's probably going to be looking like April 1st might be the date for the regenerative medicine. And then at some point later on, we'll have a conference when we're all allowed to come together and socialize. Next slide. Oh, we went down to, so I'm David Granville. I'm your host for today that will be introducing Malcolm and the speakers. So as I mentioned, the purpose of this seminar series is really to make people more aware of who's doing what within the community. So getting the people, introducing people that are more on the science side, getting them more familiar with what's occurring on the business side, and likewise trying to help people in the biotech community to get more familiar with who's doing what in the academic and clinical communities, as well as helping students to be aware of both types of careers. So our speakers here, I'll let Malcolm introduce those. So I think we can go to the next slide here. So there'll be a few housekeeping notes today. Basically, for questions and answers, if you have any questions for any of the speakers, feel free to use the chat box down the bottom to fire over any questions, and Malcolm will answer any of the questions. And please leave your microphones on mute, not that I think you have a choice. So at this point, next slide. It's my pleasure to introduce Malcolm Kendall. So Malcolm has over 3 decades in the operational management, entrepreneurial, venture capital investment, and leadership experience, the majority of which has been focused on company creation, building value in technology and biotechnology companies. He's a former venture capitalist with MDS Capital, now known as Lemira, Atyrus Venture Partners, and InterSouth Partners. And he also lectures at the Siegel Business School at Simon Fraser University, and his formative years were actually in the infantry and special forces officer of the U.S. Army. So I'm anticipating if anyone can keep us on time, it's Malcolm. So at this point, Malcolm, I'll pass the microphone over to you.
Dave, thank you. It's a real honor to be here. When you reached out and asked if I would help put this together, I was very excited. Your introduction makes me sound both old and successful, and I can confirm that I am old. I'm not going to say a lot. I'm the CEO of a microbiome testing company here in town that was founded by Brett Finley and Bill Mon. My job today is to introduce the speakers who have got a great lineup to really share with you. Bill will start with an overview of the microbiome, but then we're going to get down into some of the research that's going on here in the UBC Vancouver VGA area. And I think my hope is that if you don't know about the microbiome, you learn something about it. But if you do know about the microbiome, we push you a little bit further to show you things that are going on and how it's important and how it connects to things like the immune system and inflammatory disease and the gut-brain link and all these other things. Real tight timeline between speakers, not a lot of time for questions, but at the end we will have time where they will come back on as panelists, and we would love to address your questions. So I think as you go through, you can put questions in the chat, and they will be shared with. Without further ado, I would like to introduce Dr. Bill Mon, who is a professor in the Department of Microbiology and Immunology in the Life Science Institute at UBC. He is also my fellow co-founder of Microbiome Insights. Bill has conducted groundbreaking research on bacterial metabolism and microbial ecology. Really, everything we learned in the microbiome as it applies to the human microbiome comes from the tools and science they built from microbial ecology. And he has over 150 peer-reviewed publications. His current research employs classical, molecular, genomic, and metagenomic approaches to study bacterial degradation of steroids, and he also is focused on studying the human microbiome. Bill was awarded the UBC Killiam Research Prize and the Canadian Society of Microbiologists Murray Award for Career Achievement. And without further ado, Bill, over to you.
Okay, thanks very much, Malcolm. I'll just get my screen share in place. Okay, can somebody confirm you can see my first slide? Great, thanks very much. So in my limited time, I will just tell you a few things, key facts about the human microbiome, and share with you some of the ways that it affects human health. So for a definition, a microbiome is a microbial community associated with either a host or an environment. And we now know that virtually all plants and animals have microbiomes. And these microbiomes are overwhelmingly mutualistic with their host. And in fact, the microbiome typically performs essential functions and is an integral component of animals and plants, including ourselves. And so we've come to think of ourselves in one sense as a superorganism, not just a human individual living on its own. A really important aspect of the microbiome is its metabolic capacity. And that's highlighted by the fact that we have approximately 23,000 genes, whereas our microbiome is estimated to have about 2 million genes. And so that microbiome has a lot more metabolic capacity than we do and can perform a lot of metabolic functions which we're not able to do on our own. I also just want to mention that the microbiome includes fungi, protozoans, and viruses, which are very important, but are much less well understood than the bacterial component of the microbiome. And so most of what I'll tell you today relates to the bacterial component. So we often refer to the human microbiome, but in fact, each individual human has many very distinct microbiomes at different body sites. And so these pie charts are just showing the very high taxonomic level composition of microbiomes at different sites. As well, the microbiomes differ substantially among individual humans. And this is highlighted by the fact there's not a single bacterial species that's common to all humans. And so this individual variability really confounds our efforts to understand how the microbiome functions in the human host. However, another really important point is that while the taxonomic differences in our communities are great, the functional differences are much smaller. And so there's an idea of functional redundancy whereby many different bacterial species will perform essentially similar functions within individuals' microbiomes. And so I think to understand the microbiome, we really need to focus on the functions and the genes encoding them. And not get too confounded by the taxonomy. Okay. There we go. All right. So here are just some, at a very high level, general functions of the microbiome. And some of these we've known for quite some time. So there's digestion and the synthesis of key vitamins and hormones and protection from pathogens. More recently, we've come to appreciate that the microbiome has a significant influence on the development and regulation of the immune system. And even more recently, we've understood that it also affects development and regulation of the brain. And finally, I want to mention metabolism of drugs, because I think this is really important and needs to be better studied. So with these very distinct microbiomes among different individuals, those microbiomes in the gut will metabolize drugs differently, and I think are going to be a real factor in how individuals respond differently to drugs. So I think for anyone who's interested in precision medicine, this is an important point. So now I just want to talk about a few aspects of the microbiome as they relate to human health. And so this is a picture from what's now become a classical microbiome study. And it showed that genetically identical mice with an identical diet and differing only in their microbiome, their gut microbiome composition, could have very distinct phenotypes with respect to obesity. And so in this experiment, germ-free mice were inoculated by fecal microbiome transplants. And when the source of the inoculum was a normal mouse, the phenotype of the recipient was normal in terms of body weight. Whereas when the donor was an obese mouse, the recipient became obese. And so this was just a really dramatic example of how the microbiome can influence obesity. And there are a number of effects of the microbiome that are relevant to this. One is the digestion of recalcitrant carbohydrates that the host is unable to digest. Another is upregulation of fat storage via elevated blood sugar. And there are likely additional regulatory mechanisms impacted by the microbiome. So next I want to say a little bit about autoimmune diseases, which is obviously highly relevant to today's series of talks. And these two graphs illustrate a really striking trend, or two trends really, in developed countries during the last half of the last century. And so what we saw was a dramatic decrease in infectious diseases in these countries, meanwhile a dramatic increase in autoimmune diseases. And we now have growing evidence that there are interrelated roles of both the microbiome and environmental exposures that are helping to drive these diseases. And this is just a partial list of autoimmune diseases that are associated with the human gut microbiome. And so we'll hear about a few of these today. The important point I want to make here is that this is an association. And in most cases, we have a very limited understanding of any causal role for the microbiome in these diseases. So this is a critically important area of research to better understand this association. And this next slide addresses the question of how the microbiome could be affecting all of these autoimmune diseases. And this is one general mechanism, the influence of microbial metabolites on functioning of the immune system. And so this is from a review article, and it summarizes a large number of ways that different microbial metabolites from the gut can modulate immune system function. And so we have a rapidly growing understanding of the complex ways that the microbiome can modulate the immune system. And we're finding when we try to relate this to individual diseases, the story is typically very complex. So finally, I want to talk a bit about how it is that the microbiome can affect brain development and regulation. And this is via something that Malcolm actually mentioned in his introduction, the gut-brain axis. And so this is a way that the brain talks to the gut and the gut talks to the brain. And so it's a bidirectional communication and regulation network. And it's comprised of neurons, hormones, immune cells, and importantly, microbial metabolites. And these metabolites in some cases can directly affect the brain, and in other cases they act via the other components of this axis to affect the brain. And this axis regulates digestion, mood, sexuality, energy expenditure, stress, and other things. So it's very important. And interestingly, the axis can also modulate the composition of the gut microbiome. And so with understanding that this gut-brain axis exists, we can begin to understand why we're now beginning to find associations between the gut microbiome and many psychiatric disorders. And so here are some that I've listed where that association is fairly well established now. And again, as in the previous case, I want to make the point that this is mainly a matter of association, and we've got a long way to go before we really fully understand what, if any, role the microbiome has in driving or mediating these disorders. But in the case of stress, we do know a bit more, and we do have some evidence for an actual causal role for the microbiome. So we know that stress alters the microbiome, and so that's the association that I keep mentioning. But we further know that probiotics can actually reduce the stress response in animal models. And there are some human studies that have consistent results with this. And at a more mechanistic level, germ-free mice have been shown to have an elevated hypothalamus pituitary adrenal axis response to stress. And so this is a major mechanism of stress response in animals. And further, specific bacteria have been shown to reduce this stress response, whereas others have been shown to increase the stress response. So here we have evidence for a direct effect of specific bacteria on the stress response in animals. And so I think that pretty well exhausts my time, and I just want to leave you with a conclusion from a review. 100% of the U.S. population, and I'm confident this is also true for the Canadian population, has been or will be affected by diseases and conditions involving their microbiome. Thanks very much.
Powerful way to end. Well done, Bill. I guess we have a few minutes for questions here, if anybody wants to throw one out. If not, we will keep rolling and save them to the end. So I don't see any questions. I think we should keep moving. So it is my distinct pleasure to introduce Dr. Shirin Kalayan, who is not only a translational immunologist and a faculty member with the UBC Department of Medicine, she's also a good friend. For over 6 years, Shirin has been leading the science at QBeCo, a clinical stage Canadian biotech with an innovative immunotherapy platform that uses bugs as drugs to treat immune-related diseases and cancer. And she now serves there as the Vice President of Scientific Innovation. Her research focuses on improving efficacy and safety of therapies targeting immune dysfunction and determining how to best harness the ability of the innate immune system to overcome pathology. Finally, she has allowed me to share with you what keeps her internal microbiome and external ecosystem healthy is that she employs walking as her main mode of self-transportation. Over to you, Shirin.
Thank you, Malcolm. And thank you, Bill, for that wonderful overview of the microbiome. I think it really sets the stage here. I'm coming from this from the perspective of a translational immunologist, and the pivotal role the immune system plays in shaping our microbiome and in our interactions with it. And Crohn's disease really exemplifies when that relationship between our immune system and our microbiome breaks down. what we're trying to do at Q is really help establish a healthy relationship again with our microbiome to reinstate healthy gastrointestinal state. And we're doing this by using a microbial-based immunotherapy platform that trains the immune system to function much more optimally with our microbiome. So as a background, preventing progressive disability in most patients with Crohn's disease has really yet to be realized. We have quite a broad arsenal of immunosuppressive medication at our disposal, but we really haven't changed the bar as far as preventing that disease progression for the majority of patients. And there's accumulating evidence that suggests that patients with CD actually have innate immune defects. And I know Bill had mentioned it's often characterized as an autoimmune disorder, but it's really more of an immune insufficiency in the gastrointestinal tract that affects the mucosal barrier function. And this leads to dysbiosis and a dysregulated adaptive immune response to our own microbiota in our gut. And we have a GI-directed immunotherapy called QBECO that is designed to restore that innate immune function in the GI tract, and it's currently in phase 2 development. And I'll be sharing with you some of the interim analysis we have in terms of the clinical symptom relief we see with this approach, as well as the first signs that we are indeed reprogramming the way the innate immune system is functioning in these patients. So as I alluded to previously, the main focus of the standard of care for IBD and Crohn's disease in particular is really around symptom management. And we're really hoping that we can address the underlying problem that we see present in Crohn's disease. We know that there are many genetic variants that are associated with the disease, and many of these are related to innate immune functions, such as the recognition of bacteria and their clearance and autophagy. However, it's important to recognize that many people have these variants, but they don't really develop CD. So we know that there is a major contribution of the environment that provides a stressor or a trigger that precipitates the disease in susceptible individuals. So what we see in the healthy state, we have this really densely populated microbial community in our intestinal lumen. And there is a nice mucus layer that provides a microbial distance and a really strong epithelial barrier. The epithelial barrier is actually one of the most ancient parts of our innate immune system, and there is a feedback that provides physiologic homeostasis. What we see in IBD is that you get encroachment of the gut barrier, and you really lose this mucus layer, and the gut gets leaky, and you get essentially trespassers that leak through the gut. It's often referred to as a leaky gut. And this chronic infection is what calls up the troops for backup, and so we recruit inflammatory T cells and neutrophils, and this leads to chronic infection of the gut, and it's really hard to resolve and turn this around. As was mentioned, this is a really complex situation. What we're trying to do is really rebuild the forces here and build some muscle in the innate immune system such that we don't have these trespassers. And our therapy, QBECO, is very easily self-administered by the patients themselves, subcutaneously, similar to an injection, an insulin injection. So from our previous randomized controlled trial, which was conducted in patients with moderate to severe Crohn's disease, there were 68 of them, we were looking at symptom relief using the CDAI score, which is the Crohn's Disease Activity Index, and that's on the y-axis here. And our primary endpoint for evaluation for that study was week 8. You can see there's a nice separation in terms of a drop in the CDAI score and patients that were randomized to QBECO versus placebo. At the week 8 time point, what we did is whatever treatment the subjects were randomized to, if they were responding to that treatment, that stayed blinded on that treatment for the following 8 weeks. If you're a non-responder to that treatment, you were put on open-label QBECO. And this study design was really to facilitate recruitment, because if someone has a moderate to severe Crohn's disease and you're not going to be an active drug, you're less likely to enroll in a study. So what we learned here is that the patients who had initially responded to QBECO and stayed on treatment, they actually continued to improve to week 16. Here we can see that those who had initially responded to placebo and I should mention that IBD is one of those conditions in which the placebo rate is fairly high. It hovers around 30%. You can see that they started to lose response over time. And both the other two non-responder groups had a significant response to QBECO, more so the placebo group that were previously non-responding on placebo. So there were two main lessons we learned from this study. The first is that week 16 is, or later, is probably a better time to assess the response to QBECO. We're not really getting the full effect of the benefit of this approach at the week 8 time point. The second learning was that we need to stratify for people who had been previously treated with biologics, in particular anti-TNF agents, because they took a lot longer to respond, almost double the time that anti-TNF naive patients did. So we have built those learnings into our current study, which is focused on assessing endoscopic response, because the bar has changed for drug approval in IBD from just symptom improvement to actually an objective measure of response. So our current trial was designed with feedback from the FDA, and they said first we have to figure out when is the best time to actually evaluate endoscopic response in our patients. So this lead-in study, which is going to be completed in the next week or so, whether it's week 16 or week 26, and this will inform a larger randomized controlled trial. And we'll be looking at clinical, endoscopic, and histologic improvements, as well as confirming some of the immune biomarkers we saw that were associated with QBECO response in our previous trial. We'll be assessing the genetics and, of course, the microbiome. I was able to, quite fortuitously, enroll 2 sites in our current trial to assess trained innate immune function in our patients. Are we really changing the way the innate immune system is responding in these patients? And I was able to get some of this interim analysis in the last time. I was interested in this because our mechanism of action, if you're interested in understanding more about trained innate immunity, we had published last year the mechanism of action and the contribution of trained innate immunity in our therapy. And I'm showing to you here IL-1 data and GM-CSF levels because those are established markers of innate immune training in patients. And I looked at the response of the immune cells in patients at baseline and then after 16 weeks of QBECO treatment, and assessed whether or not the efficiency of the response of the microbiome or the microbial stimuli was increased. And even though it's small numbers, you can see they were both significantly increased after 16 weeks of treatment. What was also improved was their clinical symptom score. So here I'm showing you the pain score and the loose stool frequency in these patients. And the red line on both denote the clinical remission criteria. You can see majority of patients, as the median line, are below that remission criteria at week 26. The part that I was really excited about was this data where we had previously found that people who were good responders to QBECO had an increase in interferon gamma in their serum early on. And in this study, in my in vitro immune assessment, those that were in clinical remission for both pain and loose stool frequency were 33-fold increase in their interferon gamma production from baseline versus only 2 for people who were not in remission at week 16. So I think this really underscores the role of the productive interferon gamma signaling in QBECO efficacy. So in conclusion, so far, we have confirmed the findings of our previous trial, and that show a marked improvement in at least the clinical symptom relief of this innovative approach for the treatment of IBD. It is also the first analysis that show that we're actually changing the way the innate immune system responds. We're increasing its competence in response to microbial stimuli. And we're hopeful that this will provide an alternative for the aggressive immune suppression that's currently used for the treatment of CD, and that it may be possible to actually select patients who are most likely to benefit from this approach and monitor their efficacy while on treatment with some of these biomarkers that we're assessing. Our upcoming analysis includes endoscopic and histologic analysis from our study, and we have that within the next week or two, and I can say it looks good, and we hope to present that data in one of the 2 upcoming IBD conferences, either DDW or ECHO. And these will be analyzed in relation to the immune biomarkers I mentioned, as well as the microbiome. And I currently have Malcolm on speed dial because Microbiome Insights will be helping us with that analysis. And of course, it takes a village for drug development, so I'm really grateful to all our advisors, collaborators, and partners. And those in bold here are our site investigators, Brian Bressler at Jerry, John Marshall at McMaster, and Kenneth Atkinson at Fraser Health. And of course, Microbiome Insights has been by our side almost from the beginning. And here's a fearless team. And look forward to our questions. I believe we're leaving those to the end, but I look forward to the discussion. Thank you.
Great talk, Sharon. Congratulations on all your success, and hope these trials, continue the success. Stuart has a question, actually.
Sure.
Can you tell us more about what is in the QBECO?
Yes.
Chime in here if I missed it.
Yeah. Oh, that was going to be part of my discussion, but I realized I went over 10 minutes when I went into it.
We're doing okay on time.
Excellent. So QBECOs, we have actually an immunotherapy platform called site-specific immunomodulators, and they're derived from bacteria. They're actually endogenous and a common source of infection in the target organ. So QBECO is derived from a clinical isolate of E. coli, and that's why when we sort of administer it subcutaneously, the immune system, we found within a few hours tries to hone on where's the greatest concentration of this bug from and sends its army there. It's a quite brilliant way of targeting specific areas of the body and focus the innate immune system there.
And we have a question from Rachel. Have to profile the patients receiving QBECO for 16S composition. How much can you attribute to the treatment itself versus the potential downstream outcome of modulating the composition of the gut microbiota?
So we haven't really assessed changes in the microbiome yet. That's what Malcolm's job is in this study. We haven't, it's not a part of the criteria, but we want to understand more on the level of dysbiosis that's present in Crohn's disease and how that changes. What we often see is the opportunistic bacteria that thrive in the presence of inflammation. They're often anaerobic in nature, are elevated in the context of dysbiosis in the gut. And what we want to see is change in that the diversity and the functional characteristics of the microbiome with treatment. And so we still need a lot of work to do to understand what is a healthy gut and what is that's associated with disease.
So I'm going to jump in here with a question before we throw Stuart up there. The microbiome, dysbiosis of the microbiome, has been demonstrated to be part of the disease cascade in IBD. I'm just curious, with your therapeutic approach, obviously you're targeting the immune system, but does that have an effect on the gut microbiome? How does that work?
That's an excellent question, and a lot of the work we had to do on that aspect was preclinical. So we were working with Bruce Valence at BC Children's, where Stuart is from, and we were using a MUC2 mouse model. So these mice get a colitis quite spontaneously, and what we found is by improving the immune resiliency of the gut, which included upregulation of specific types of defensins and antimicrobial peptides, these target specific types of bacteria. They favor actually the good bacteria, the lactobacillus, and it gets rid of the more unfavorable type of bacteria. And so that's how the immune system interacts, because it chooses who is friend and foe, essentially, and shapes the community. I've done previous work, if you're interested, you can look it up, in the oral microbiome and adverse immune drug effects. And what was striking was the main predictor of the composition and diversity of the oral microbiome in these patients was really how strongly their immune system responded. So if they had high levels of rank ligand and TNF and aryl hydrocarbon receptor, then their their oral microbiome tended to have less streptococcus, for example. And so that's how the immune system really helps shape what's there and how much of it is there.
Very cool. Great answer. So with that, we're going to move on to our next speaker, and it is an honor to introduce Dr. Stuart Turvey, one of my favorite people. He is a professor of pediatrics at UBC, where he holds both the Canada Research Chair in Pediatric Precision Health and the Aubrey J. Tingle Professorship in Pediatric Immunology. He is a clinician scientist and pediatric immunologist based at BC Children's Hospital. His research program is focused on determining the early life origins of asthma and allergic diseases, as well as harnessing the power of genomics to diagnose, treat, and prevent pediatric immune disorders. And I truly hope, Stuart, you're going to talk about the child cohort study. And so now, over to you, sir.
Okay. Malcolm, thank you so much for that kind introduction. So I chose a more provocative title than I think I was given, and wanted to talk about dirty diapers and think about the relationship of the gut microbiome in early life and asthma as one key example of an inflammatory disease of childhood. Okay, there we go. So I'm a pediatrician. These are the sort of patients we look after, and we, in pediatrics, really believe strongly in the developmental origins idea that many of the health challenges begin very early in life, and so understanding these early life events will help us potentially intervene to prevent long-term consequences. And the disease I want to talk about today is asthma. Asthma is a classic inflammatory disease of childhood. Bill Moen showed that this is one of the diseases that has increased so substantially in the last decades. And as a pediatrician, this is one of the main reasons children access the healthcare system, come to hospital, or miss school. So it's a really burdensome condition. And there's been decades of epidemiological studies really trying to understand why do some people develop asthma. And here is a 2006 review article just trying to summarize some of those. But as we go through those factors and look at some of them, one unifying factor is potentially the microbiome. So the microorganisms that live with us and on us, that are influenced by our exposure to farms, the family size we're born into, whether we go to daycare early in life, whether we receive antimicrobial therapies. And so it made sense to start looking at the microbiome as a potential unifying factor in what's been proposed as the hygiene hypothesis, the idea that one of the reasons that allergic disease, including asthma, has become so common is that we're too clean these days. And so to start that journey, which was a daunting journey, more than a decade ago, we launched a study called the CHILD study. And the CHILD study is a study launched across Canada that includes about 3,500 families recruited in 4 sites, including here in Vancouver. And we've followed up those very generous families now for almost 8 years. We're finishing the 8-year visits, but the families stuck with us at a retention rate of more than 90% out to 5 years. And one of the inspired things we did really was to collect biological samples very early in life, including stool samples from those dirty diapers. And that's given us a lot of information about the structure of the microbiome in early life and how that links to later health outcomes, including how it links to asthma and allergic disease.
I want to speak briefly about a paper that was now published some years ago in 2015 that was a partnership of many investigators, including with me and Brett Finley and many child study investigators. And in this study, we looked hard at the structure of the microbiome using 16S sequencing. And what we noticed was that some of the rare taxa seemed to be depleted in the children who went on to develop asthma. And interestingly, that depletion was seen in stool samples at 3 months of age, but really wasn't there in the stool samples at 1 year of age. When we dug into this more carefully, the taxa that emerged, we nicknamed flavor, because me and many of us struggled with the complex Latin names, the Faecalibacterium, Lachnospira, Vianella, and Rothia. And what we found was that this group, the flavor gang, seemed to be at lower levels in the 3-month stool sample of the children who had the highest risk of asthma. This is a lovely correlation, as Bill pointed out. But one of the strengths of this study was that Mary Claire Arietta and Brett Finley's lab were actually able to do mechanistic experiments in mouse models, showing that deliberate supplementation of the flavor organisms protected from the airway inflammation in these mice models. And so that opened the idea that perhaps we could intervene in early life in humans to replace some of these missing microbes. We were delighted that this work has subsequently been replicated in different cohorts around the world, really, I think, suggesting that there's a significant and biologically plausible and established link between the early gut microbiome and later risk of asthma and related allergic diseases. So one of the weaknesses of that first study is that we didn't consider what came first. We didn't consider what might change the structure of the microbiome such that some children were at increased risk of allergic disease and some seem to be more protected. The study wasn't designed that way. But using child study, we were able to start to ask that question. And so we undertook that study with a specific focus on a classic modulator of our bacteria and our microbiome, and that's antibiotics. And so many studies have identified associations between early life exposure to antibiotics and subsequent risk of asthma. But there's still, despite those many associations, there are many unanswered questions. One obvious question would be, do these associations still hold true? We're being so careful with antibiotics and antibiotic stewardship. Is it still true? There's a risk of confounding by indication in the way the antibiotics in relation to the wheeze, and we didn't know any of the mechanisms that were at play. And so this paper was just published, I guess, almost a year ago now, in 2020, just as the pandemic kicked off. And it was a really lovely collaboration with Brett Finley and the child study group, but also with David Patrick and people from the BC Centre for Disease Control. And here's what we found. So David Patrick's group were able to look at provincial administrative data, looking at prescribing practices from 2000 to 2014, and looking at the rate that antibiotics were prescribed to children less than age 1. You see a really nice decline there in the blue. And then they were able to look at the asthma diagnosis in this same cohort for children diagnosed with asthma from ages 1 to 4. And what you see that after some delay, the red line also starts to decline, really nicely paralleling this fall in asthma prescription rates. So that was, I think, a nice, intriguing association. I want to say the numbers are striking, so this way it's easier for me to understand it. So in 2000, about 70% of kids were given at least one course of antibiotics before their first birthday in British Columbia. By 2014, that had fallen in half. So it's only about 30% of children. And there's still a lot of way to go in terms of reducing this exposure that was almost ubiquitous previously. In the child study, we were then able to see if this population-level observation held true for individual kids. And indeed it did. So we were able to look at children who got antibiotics in that first year of life and see what their rate of being diagnosed with asthma at age 5 was. And what we saw was that having a course of antibiotics before age 1 was associated with doubling the risk of being diagnosed with asthma at age 5. And this was after adjusting for classic confounders. There was a nice dose response as well. So only about 5% of children who received no antibiotics ended up with a diagnosis of asthma, while for children who received 3 or more courses of antibiotics in that first year of life, the rate of asthma diagnosis increased to 17%. Not surprisingly, we were really interested in what microbiome features might be associated with that. And here we use 16S sequencing, which is not great at resolving down to species level, to look at what might be going on. And in the study, we showed again that in the children who had asthma and had received antibiotics, we saw depletion of specific taxa, with really essentially all being depleted except for one. And this list is becoming really familiar group of taxa, Faecalibacterium and Ruminococcus, for example, are classic organisms now that are known to produce short-chain fatty acids, some of those metabolites that Bill Moen introduced that are so essential for training the immune system. And then we were able to use structural equation modeling, a statistical technique, to try and understand the link between antibiotics and asthma. And using this statistical technique, we see no direct link between the antibiotics and the ultimate asthma diagnosis. But when we included the structure of the microbiome as a latent variable in that modeling, we saw a significant indirect effect, suggesting, as we would have hypothesized, that the antibiotics seem to modulate the structure of the gut microbiome, and it's through that modulated microbiome that we're starting to see the increased risk of asthma. So we smiled, we were happy with these results, but Bill's concern rings in our ears, and I think we absolutely acknowledge that this work is correlation and needs to be replicated in other populations, and then we need to dig into the mechanisms that underlie that. Perhaps the metabolites that the microbiome is generating that trains the immune system and modulates the disease. But I'll conclude by saying from this, we still see very high rates of antibiotic exposure, even in contemporary Canadian children. 1 in 5 Canadian children receiving a course of antibiotics before their first birthday. We see that this course of antibiotics is associated with essentially doubling the risk of being diagnosed with asthma at age 5, and that the antibiotic exposure is associated with dysregulation or dysbiosis of the gut microbiome and changes in the developmental trajectory of that microbiome. I think this knowledge and starting to name and identify the organisms and taxa and their metabolites has the potential to allow us to intervene potentially in children who absolutely need antibiotics. There may be a way to rebalance that microbiome. But I think it's also interesting to think that a significant reduction in pediatric asthma may just be an unexpected dividend of prudent antibiotic stewardship. So I'll end by acknowledging the huge crowd of people related to the child study who helped us do this work, and some of the funding agencies. Thank you.
Thank you, Stuart. Always impressed, and as you know, I'm a big fan of the child study, and a believer that the one place that the microbiome may have the biggest impact is on the early life microbiome and how it relates to sort of the priming of the immune system and health and the potential for chronic diseases. So don't stop what you're doing.
Thank you.
Regretfully, we don't have time for questions because we're right on time again, and Dave insisted that I use my militaristic training to keep things on track here, so we're going to keep moving. Last but not least, and I love her, Dr. Suzanne Lewis, who is a clinical professor in the Provincial Medical Genomics Program at BC Children's Hospital and UBC. She's also the Chief Medical Officer and Vice President of Research at the Pacific Autism Family Network and Director of the collaborative iTarget Autism Project, which I hope she's going to talk about. The iTarget Autism research is focused on subgrouping the autisms via whole phenome, genome, metabolome, microbiome, and proteome discoveries with a precision medicine focus on cause, not only symptoms. Suzanne's contributions to the autism community and to research, science, and technology have been recognized by a 2019 YWCA Women of Distinction Award and a 2020 Top 100 Canada's Most Powerful Women Award from the Women's Executive Network. Thank you, Susan. Over to you.
Great. Everyone hear me okay? Thank you very much for that, Malcolm. And thank you, everyone. I'm very much enjoying the presentations and really appreciate being invited to this event. And I'm very happy to talk to you a little bit about the work that we're doing in the area of the autism microbiome and the iTarget project, which Malcolm nicely explained. It's a name for individualized treatments for autism recovery using genetic and environmental targets.
And Suzanne, you're in presentation mode. We can see your speaker notes in the next
Oh,
slide.
gee, okay. Well, we don't want that. So let me just see how I can get out of that. Oh, I thought we had stopped that before. So let me stop sharing, and we'll just go to a big screen, which I hope. See, maybe this is the one I want to do. Go from the beginning. My apologies. Here we go. Let's see if this works.
Perfect. Success.
Okay, we're good. That's this. Okay, is that okay? Or are we back to speaker notes again? Is that okay?
Perfect.
Perfect. Okay, great. So let's start just by saying, well, what is autism spectrum disorder? And we know it to be a complex neurodevelopmental disorder. It's characterized by impairments in social communication, primarily in reciprocal interactions. But it also is known to show concerns, deficits, illustrative of repetitive, somewhat rigid, and restricted stereotypic behaviors. In Canada, according to the Public Health Agency of Canada, autism affects 1 in 66 children, and it's about 1 in 54 if we're looking at the CDC in the United States, and it's 4 to 5 times more common in males. Where females are affected, it suggests a more stronger genetic liability. And the numbers are increasing, of course, because the prevalence is increasing, as shown by these numbers collected from studies through the PHAC in the East Coast. And what's concerning is that the average age of diagnosis in BC, this comes from Ministry of Child and Family Development, is still quite late, only 6 to 7 years. And what's concerning about that is that we miss this very important window of opportunity before age 6 to 7 years, where the earlier the diagnosis can be made and the earlier intervention starts, we know that this impacts upon a much better outcome and more positive developmental trajectory going forward. So that's our interest in looking at other tools towards earlier diagnosis that can help inform earlier intervention. So what causes ASD? ASD is a multifactorial disorder. We know it has both genetic and environmental influences, specific clinical genetic syndromes like Fragile X syndrome or Rett syndrome you might have heard of, or changes in chromosome in terms of duplications or deletions, or copy number variations and single nucleotide variants have all been implicated in autism. And really, I think in terms of differentiation, we know that about 20 to 30% of cases of autism are due to rare gene variants, but of large effect, and that's primarily your copy number variant and your single nucleotide variant. Whereas for the more common cases, the majority of cases, in the 70–80% of those with autism, the causes are unknown because they're due to more common variations of very small effect. And some of those variations, too, may not necessarily all be genomic, but also environmental, such as maternal congenital infections or drugs such as valproate use if moms have epilepsy or alcohol exposures during pregnancy. So I think what's really important to look at and what we can say is that autism really is more commonly an epigenetic interplay between genetics and environment, where I believe one of the most exciting fields to emerge within the environmental domain for autism is certainly the effect of one's own commensal organisms in microbial environment or the microbiome. And a lot of this information has been emerging over the last 10 years or so, as Bill showed, that each area of the body has quite a distinct group of microbes. The area circled in red here is that for the colon, where there are greater than 1,000 species in the human GI tract, and the microbiome outnumbers our own genome by at least 100-fold. And it's also, showing here where in the colon, the differences are different from different parts of the body, but in the colon, in order of commonality, we see more of the Firmicutes, the Bacteroidetes, actinobacteria, and proteobacteria. And these microorganisms and their neurometabolites can impact functions of the microbiome, which has been touched upon in the other talks, such as the catabolism of undigestible carbohydrates, the stimulation and development of the mucosal immune system, as well as short-chain fatty acids, which are interesting to look at, because often we can see these in the urine of individuals, and they're foreign to humans. They're made by microbiota that humans don't make. So it's an interesting way of looking at this impact. And some of those short-chain fatty acids are really important to intestinal integrity. Butyrate would be one of those as well. So, why even consider the role of the gut microbiome in autism? And, there are findings that point to the gut playing an important role, and the kind of clues that we see towards that is that there's still a very high prevalence of gastrointestinal symptoms in children, at least 50%, some as high as 70% in cases, some a little bit lower, but 50% on average. 85% of kids with autism have constipation. And what's interesting, too, is that the more severe the GI symptoms, we often see a much more severe type of autism. And some of those autism subtypes are even regressive in nature. So that's an interesting subgroup to look at. We find where there are gut abnormalities that there's also a coexisting history of developmental regression. Now, this also has been talked about a little bit, where the gut-brain axis is the bidirectional communication and interaction between the GI system and the central nervous system. For autism, it's particularly relevant about how it influences the central nervous system, and its influence on the gut is through a number of different complex means, through the autonomic nervous system with the vagus nerve in particular, the hypothalamic pituitary axis, as well as the enteric nervous system as well, whereas the role of the gut up to the brain can involve gut neurotransmitters, microbial metabolites, immune activation, and cytokines. And the metabolites in particular are, as I said earlier, are foreign to humans in many cases. And when they go to the brain, this open highway called the gut-brain axis, they're recognized as foreign and can mediate sort of the migration of microglial cells, inflammatory cells in the brain, and other related processes there that create a neuroinflammation that certainly is an important involving area of etiology for autism. It's one particular important subgroup that I think is reflective particularly of these kids with GI problems. Now, the gut microbiome can be impacted in many ways, by diet, environment, genetics, age, and sex, but it can also be altered, very early on in life. For example, if mom has sort of an unusual or, colonization with bacteria, often if this is an in utero environment, there can be epigenetic effects on the developing brain. And there have been studies that have shown that the microorganisms and actually the neurometabolites can impact gene splicing in the brain of a developing fetus. Of course, there's been consideration as to differences between children who go on to have autism having a higher rate of their moms having had a C-section. They see a different colonization of bacteria than if they pass through the birth canal and normally colonize themselves in that way. It's not unexpected to see differences if kids are given treatments early on, as Stuart was talking about, including even probiotics, or if they breastfed versus take formula, which also lead to different colonization of bacteria. And all of these things, can impact, how the microbiome can impact, things going forward is that it affects nutrient uptake, mucosal-associated immune response, and also levels of vasoactive hormones as well. So there's been a lot of studies going on in recent years. There wasn't so much last year. This is, a summary of some of the larger clinical trials. There's others still ongoing, I'm sure that we'll hear about. But these types of studies looked at, of course, differences in the microbiome of children with autism versus those without, and what, what they might see. And the two areas on the right here, the two columns, show that in some children, the various microbes were increased, and in other children they're decreased. But if you look between these studies, there's really no consistency there. And some of the reasons for that inconsistency might be that there were different age groups involved. I think that plays a significant factor. Maybe since these studies are from around the world, there's different environments and access to different types of foods being taken that might impact that. Or some of the studies looked at sibling controls, matched for gender versus more typical population controls matched by age and sex. And so, all of these studies, though, did look at changes in diversity, where alpha diversity is looking at differences between the species seen in those with autism, and beta diversity is that due to differences in environment. And the trouble was that almost all groups found altered diversity, however, it always wasn't in the same direction. So iTarget decided to look at the microbiome as one of our key environmental factors to associate both with the deep clinical clinome and phenome findings that we've been collecting on children for a couple of decades and databasing that. We have whole genome studies for our kids. We have now integrating the metabolome through work with Molecular U, and of course with Microbiome Insights, the microbiome, and the proteome as well. And the whole thing is aimed to try and find insights into the cause of ASD that can tell us also what's going on at the brain level, and perhaps coming up with some earlier diagnostic tools. So the study design is pretty simple. We have 1,600 subjects that are all up and ready to be tested. They're selected on the basis of having a history of GI issues, but not a known organic GI disorder. They're excluded if that's the case, or if there's no control family member, or if they're already taking pre or probiotics. We siphoned down to a smaller 50-subject study, and the work of Dr. Sarah Redmond looked at just relative abundances of different types of bacteria. But prior to doing that, we had a pre-collection questionnaire, and the sample collection was assisted through Life Labs for us, and the packaging of the kits was assisted through Molecular U and Microbiome Insights. And the questionnaire is helpful for a variety of reasons to know a little bit about each participant. Those factors are listed here. The maternal pregnancy history, childhood history, medication history, GI conditions, food allergies or exposures to the different environments, like Stuart was talking about, even daycare centers, travel, of course, now being a factor in the area of COVID, et cetera. So we had all that information, and the actual collection involves blood, looking at 131 metabolites and 137 different proteins through the Molecular U collaboration that we have that are studied through the Metabolomics Innovation Center and BC Proteomics Network. The urine is looked at for 73 different metabolites in the stool, of course, with the help of microbiome insights, looking at 16S rRNA, amplicon sequencing, shotgun metagenomic sequencing, and diversity and abundance of microbial species plus metabolic potential. And all of these studies were controlled by a same-sex family member, ideally a sibling. And just in generalities here, because this was a small study, the persons with autism were found to have a similarly altered microbiome profile. It's actually surprisingly consistent, showing an increased abundance of the Bacteroidetes phylum, showing a significantly decreased abundance of Firmicutes that can include Clostridia, Lactobacillus, and Faecalibacterium, as well as the significantly increased Bacteroides to Firmicutes ratio greater than 1.5. What was interesting is we did have a pair of monozygotic twins where both twins showed a consistently abnormal microbiome profile, but there was the microbiome profile of a more severely affected twin that also showed later onset regression, also showed the more severely altered microbiome profile. So just very briefly, we're starting to see a unique microbiome profile emerge. And once we're able to expand this into more studies through funding recently received, hasn't been announced yet, so I'll keep my tongue to myself. I won't say anything. But anyways, we hope to do a greater study and look at the implications in the future for the microbiome perhaps being helpful for more targeted and early diagnostic testing. Certainly helping to inform treatments that can go beyond behavioral interventions alone, which is all that's available right now. Look at prebiotics, probiotics, and the gut microbial transplants even as a possibility. And even in the course of management of a child, perhaps averting some of the more challenging behaviors in ASD where behavioral interventions don't work. And we've actually had some more anecdotal success in this area, particularly in the realm of self-injurious behaviors, but also ADHD and improving expressive language function. So I'd just like to stop there and say thank you to everyone involved and for your attention. I'd like to thank all of our participating families who've been with us for more than 20 years now. They've been very dedicated, allowing some longitudinal assessments. My great team, some of them pictured here, the work of Francesca Bell-Peters and as well Sarah Redmond, who Francesca is a third-year medical student at UBC now, and Sarah's postdoc now off at Sick Kids training to become a medical geneticist. Steve Shearer has worked with us for the whole genome, just together with Steve Jones. And of course, I'd like to thank Microbiome Insights and the folks there, Molecular U, and those helping us at the Metabolomics Innovation Center, and our funders as well. Thanks very much for your attention.
Suzanne, very impressive. Thank you. That's a remarkable study, and I know there's a lot more to come there. With that, we have exactly 17 minutes for Q&A. And I'd love for all the speakers to come on to the panel, and I think these questions will be addressed to all of you. We have some questions already in the chat box here, and I will jump into them. Some of them are more specific. We will start with, hold on, I'm scrolling backwards here. All right, I think this goes to you, Stuart. It's here's one from Sharon. And Sharon, the panelists, you can grill each other, you can support each other, you can argue about things. Let's make this fun. Do you see a sex difference in asthma development in children? Is the first question for you, Stuart.
Yeah, absolutely. And there's a sort of complex time component as well. So in preschool asthma, it's boys, male sex, and then there's this puberty time switch with females becoming more common around puberty. So yeah, there's a big sex component, and we work hard to always include that within our analysis because you miss things if you lump them all together.
And the next question is from Natalia. And Natalia, I think we're talking tomorrow by phone. I'm looking forward to that. And I like this question. It's what other diseases were analyzed in the trial study that may have been related to the microbiome? And this is important.
Well, Malcolm and Bill and Suzanne and Sharon and I would tell you that many, maybe all diseases have some microbiome component. And so I think one of the great things about child study is we called it child study for a reason, Canadian Healthy Infant Longitudinal Development Study, because it was a pretty holistic look at health and wellness. And so we can look at simple things like anthropometrics, height and weight, blood pressure, but then I think neurological functioning, school performance, and then early origins of cardiovascular disease and mental health are other things that we're beginning to look at, and I think you may have a real microbiome link. So yeah, we're excited about doing all of those.
It's an incredibly rich data set. I think your career is taken care of. You won't have time to work on anything else.
Thank you so much.
No problem. The next one is from Dave Granville, and he gushes a bit here. He says, "Thanks to all the speakers, exciting work." That's Dave for you. But I really like his question. He goes, "I was wondering if you could comment on where we are at, how far away with respect to taking this knowledge on the microbiome and therapeutic applications, such as designing targeted antibiotics, i.e. against bad microbial populations, precision, prescription, custom probiotics to augment good microbiome, and other approaches." Now this is, I would like all the panelists or speakers to talk on this, and just because you're the closest to me on this damn Zoom screen, Sharon, do you want to start?
Yeah, it reminds me of a quote by Alan Watts who said, "The road to hell is paved with good intentions." And I think that you don't really have sufficient clarity of given the complexity of the interactions between the hollow genome, which is essentially our microbiome and our own genome and how they interact for a given individual. And I think selecting out one or the other could, be sort of like a dominoes effect. And I think we need to understand the big picture before we can really move towards looking at single therapies. I think this, and I think that's why we have taken a more approach that we're retraining that arm of the immune system, as was spoken to the hygiene hypothesis. It was evolutionarily, this stimulation by the microbes is what trained our innate immune system. When you take it away, that training, either through antibiotics or a very sterile environment, we're living in the time of COVID, and I find this to be a really fascinating period because post-COVID, after being isolated, what is going to be the consequence? What I'm really interested in seeing of this sort of change in the exposure to the microbiome and their stimulation. So, that's my take on it.
There are days I feel like I'm in a petri dish. I don't know about you. So, Bill, over to you, but there's a second question that Dave threw out there. He's trying to look smart here. He says, thoughts on microbiome profiling as a predictor risk factor of future disease. So you get to both of those questions or thoughts or things that Dave wants to know about. And Suzanne and Stuart, I'll come back and let you talk about this as well.
Sure. I will jump right to the profiling question. So I've been involved in a number of studies where we've tried to do that. I think there's a lot of potential. One of the things that's interesting is that often you can use machine learning and come up with strong predictions with no real understanding of the underlying, structure that's actually might be, linking the disease to the microbiome. I think, one of the issues that I touched on in my talk is the variability among individuals is a real confounder here. And I'm personally very interested in pushing the profiling of metagenomes rather than just taxonomic profiles of microbial communities. I think we're going to find a much stronger correlations between the overall gene content of the microbiome and diseases than the taxonomic profile. And I think also that may kind of overcome the issue of, or at least partly overcome the issue of variability among individuals.
Yeah, I'll jump in there if that's okay. I totally agree with Bill on that. That the metagenome was helpful, and looking at sort of the metabolic profiles as well that these bacteria are showing in the studies, not purely the 16S RNA studies or just looking at sort of relative abundance levels. I think if we get a clearer idea of what's happening metabolically, and we can even integrate those findings with other factors as well, because we're all dealing sort of with complex genetic diseases linked to genes and environment. Where it might kind of culminate as all of these factors culminate is in sort of a metabolic profile that would point to the specific bacteria phyla involved. So I think it's got to go both ways. I'm not seeing it right now because of the variability between people for it to be purely microbiome, although we had some pretty cool results with the autism study. But I think what's emerging too is to look at what's happening at the metabolic level as well, which I know Microbiome Insights is doing as well.
Stuart, thoughts?
Sure. So in the asthma and allergy space, there are clinical trials, early phase clinical trials, delivering consortia, so groups of live organisms, seeing if you can replace missing organisms, repair dysbiosis. And that's started, and I think those studies will be ongoing. But I also think there's things we can do today. And I think there's sort of public health messaging that comes from our work. So for me, let's do a good job at looking after a kid's microbiome, and those messages would be avoid antibiotics unless you absolutely need them, encourage and support vaginal delivery, encourage and support breastfeeding. These things are things that are good for child health, for women's health potentially, and we can do them to help promote healthy microbiome. We can do those today without needing clinical trials. So I think there are messages
and get a dog,
for
right?
us today.
Get a dog.
Get a dog and eat a broad diet, and if Brett Finley were here, he'd tell you to let them eat the dirt. So, these are things.
He's crazy. Yeah. Okay, the next question is on ASD, so this is going to be for you, Suzanne. ASD has been linked to improper dendritic pruning by the microglia in the brain. Our enteric nervous system is considered to be our second brain. What impact do you think improper pruning / development of the enteric nervous system could impact the GI symptoms that are observed in ASD, as well as potentially shape the gut dysbiosis observed in ASD?
Oh, that's a big question. I'm not one that has looked at the brain sort of structure. I would love to do that. With folks, that had some work involving the neuroscience group in the life sciences building with Srinivas Bamji. Yeah, I mean, sure, there's lots of things that microglia can do, particularly in terms of affecting brain function. It may very well affect brain structure as well. But I'm afraid, there's just not enough information for me to answer beyond being just purely speculative of what it might do, at the enteric nervous system level. So,
Suzanne, it sounds like you might need to collaborate with Rachel Horn, who asked that question.
Yes.
That's a really
absolutely.
good question.
Yeah, it's a really good question.
I'm glad I didn't have to answer it.
Yeah, we'll also collaborate with Rachel for sure too. We should.
There you go. The next question is from Helen from what? Hello, Helen. How you doing? Thanks for highlighting the important issue of selection of controls in microbiome studies. The speaker alluded to issues with selecting siblings. It would be great to hear a little more on this issue and what the pros and cons are of the different types of controls one should consider. And that is a very good question. And I'll open it up to all the panelists. That's you all may have experience with that.
I could jump in and say, I think something that's powerful is actually longitudinal sampling. I think one shot, one day doesn't tell us a lot. So I think some sort of longitudinal sampling is powerful. And then, yeah, the controls are hard. I think it depends on the analysis. I really like Suzanne's setup for using the family members and to try and control for diet and all these other things that are so hard to control for. The way we try and get around it in child study is having fairly large sample size that can help. But my one encouragement would be, I think the longitudinal sampling is really powerful.
Bill, any thoughts?
Yeah, I would.
Rodents, there's a cage effect.
Yeah, I would partly echo what Stuart just said. I was involved in discussions about setting up the autism study, and the issue is you have individual variation, so large sample sizes really help with that in order to increase the probability of finding significant trends among the groups you're comparing. And the beauty of including family or using family members is that you at least you balance the environmental effects to some extent. So half of your controls will have a similar range of environmental conditions. Unfortunately, they're not like mice where they all had virtually identical environmental conditions, and so obviously that's one reason why animal experiments are so powerful.
All right, I think that answered that question. That was a really good question, Helen. Thank you. And I love this last question. This is a great question to end the panel on. From DRMDMC, not sure who that is. It sounds like a rap band or something. Thanks for great presentations to the speakers, all of you. Knowing associations of gut microbiome with health and disease, what do you do to optimize your own microbiome? Suzanne, you want to start?
Yeah, no, I'm a big believer. Absolutely. I honestly think that Western diet has an influence on our microbiome. I think that's why we see sometimes some differences geographically across the world in terms of incidence of autism. I honestly do believe you are and behave sometimes by what you eat. So I try and follow a more paleo diet and avoid processed foods and all the high glycemic foods that the more culprit organisms tend to thrive upon. And I also take daily prebiotics and probiotics, I think that there's some value there. So yeah, others might disagree, but I think it's a good thing, and I feel good for it.
Sharon, how about you?
I think you shared my secret, but I'm also lax with I'm also lax with the 5-second rule. When your food drops, just let it sit there for a while.
I agree too.
All right.
I
Go ahead.
agree in terms of having a healthy diet. I'm mostly pescatarian and eat a lot of different types of fibers coming from different types of vegetables every day and all that good stuff. But I also think the stress control, the cortisol levels, and getting sufficient sleep and exercise is critical, and we don't pay enough attention to that.
Okay, Stuart, we're running out of things here.
Yeah.
Surprise us.
Drink beer. That's the answer.
It's fermented beverages, of course. I knew that. And Bill?
Well, I guess I'm afraid I'm not someone with the key to success. So I do a lot of the right things. My diet is is pretty good. I exercise. My sleep pattern has gotten pretty stable during COVID. But I don't have a tremendously happy gut. And I think I'm probably an example of, you know, this idea that the early development of the microbiome kind of sets you up for life. And I'm kind of think that I'm somebody that it's too late to really improve things that much. You know, you can tinker with things at the margin, but
we'll figure it out, Bill. We'll figure it out. And for me, it's kefir and granola in the morning, everything in moderation, a Mediterranean diet, and that's about it. It's 4:25. Mic drop. Back to you, Dave. I did it.
Well, thank you, Malcolm. An outstanding job chairing, despite what they say. I also want to thank all the speakers. It was an outstanding session, lots of great discussion, as well as thanking Kara, Amanda, and Vivian for putting on these events every other month as well. They did a great job. And lastly, for anyone who wants to suggest future topics for conferences or for the sessions, take a picture of this QR code here. It's kind of neat. Take a picture, and then it actually pulls you up to this question, and you can actually rate the relevance and contents of this session as well, and what's going on. And with that, yeah, I'd like to thank all the people that asked questions and all the speakers, and have a good rest of your evening. Thank you.
Awesome. Great job, everyone. Thank you.
Good job.
thank you.
Yeah, thank you, everyone.
And take care.
Bye.